So what I'd like to do then is give you just a couple of little vignette snapshots of some of the kinds of things that we think about a lot in my laboratory. And we spend a lot of time thinking about viruses in the ocean primarily, but as a reservoir of genetic diversity and as a major driver of global processes. So I'm not going to, hopefully I'll go quite quickly, but hopefully it's not too complicated to follow these ideas. And so there's really only three things that I want to touch bases on today, and one is that there's beaucoup viruses in the ocean. There's lots and lots and lots of them. That they're major players in global biogeochemical cycles, and that we have this vast reservoir of diversity that's essentially unknown. And after listening to some of the talks already this morning, I think I'm going to go out there and see if I can find the origin of retroviruses in the ocean. So, and if you look at a slide like this, it's interesting. This always reminds me of something like the Hubble Space Telescope or something, but we just don't get the same kind of publicity. I suspect our budget isn't quite as large for publicity. But what this is a drop of seawater, essentially, which is stained with a fluorescent dye which binds strongly to nucleic acids and has a very high fluorescence yield. And in fact, all these little dots that you see are actually viral particles. And so that's kind of what seawater looks like. It's full of virus particles. And that's something new. About 20 years ago, we really didn't know that there were viruses in the ocean. But in fact, now we know that on average, there's more than 50 million viruses in every milliliter of seawater. So every time you go swimming, by the way, you're swallowing 500 million or so trillion viral particles just from the amount of water that you bring in. So it's good to know that swimming is quite harmless, so most of these viruses are going to be harmless to us. But it really changed our perception in terms of what's going on. And so one of the things I thought about, well, you know, if there's a lot of viruses in the ocean, to kind of keep this planetary theme going on here, is just one day when I should have been writing a proposal or something, I said, I wonder what would happen if you took all the viruses in the ocean and just strung them end to end. How far would they go? And I thought, well, maybe they go to the moon, or maybe they go to the sun. So it's fairly easy to do. I mean, we know the volume of the ocean, and if you just take the minimum amount of viruses we find anywhere in the ocean, which is about 1 million per mil, then, oops, I went backwards, I'm sorry. We know that there's about 10 to the 30th viruses in the world's oceans, which should be showing up there. And so 10 to the 30th viruses, so that's a big number. I don't really know how to think about that. So if we string them end to end, we know if we take the average phage length, and most of the viruses in the ocean are bacteriophage, that's about 100 nanometers. So we end up with about 10 to the 23 meters. And that's still a big number. That's larger than the US deficit, for example. And so it's very, very hard to kind of get your mind around. So I thought, well, let's divide that then just by, let's divide it by kilometers. And of course, we're still up there at, what, 10 to the 20th kilometers. Well, let's just divide by light years, right? 10 to the 13th kilometers. That gets us. So if you stretch those viruses end to end, they would go 10 to the 7th light years. And just to put that in perspective, Proxima Centauri, which is our closest star, is about 4 light years away. The Crab supernova is 4,000 light years away. Our own Milky Way galaxy is about 150,000 light years across. So in fact, those viruses, if you stretch them end to end, would go further than the nearest 60 galaxies. Okay, and so certainly, hopefully, point one, I've convinced you there's lots of viruses in there. But it's more that it's not just a trivial exercise, but you have to think every one of those virus particles, in fact, contains genetic information. They're under evolutionary pressure. Every time they infect a cell, there's an opportunity to transfer genetic information to that cell and drive evolutionary processes. And again, to give you a big number, in the ocean, there's approximately Avogadro's number of virus infections per second. About 10 to the 24th viral infections per second. So even if things are pretty rare, they're still happening all the time. And so I think when we start thinking about viruses as being drivers of evolution and all of these sorts of things, there's lots and lots of opportunities. So then the other thing is, this really kind of changed it. You can look at it another way. You can think about biomass. So it's about 200 megatons of carbon in viruses in the ocean, which is equivalent to about 75 million blue whales. It took me quite a long time to find the carbon conversion factor for blue whales, but I did. And you know, in contrast, if you look at all the whales that ever existed in the ocean, it was only about 13 megatons, right? So viruses, just to give you an idea, they're a significant component of the biomass, but they're absolutely dwarfed by the prokaryotic biomass, the bacteria and the archaea, which is about 5.2 gigatons. And so that brings me to my next point here, which is essentially the oceans are microbial, and I think this is really, really important to keep in perspective. About 90% of the living biomass in the world's oceans, if you could weigh it all, is prokaryotic. And if you throw the protists on there, that comes up to 95%. So that 95% of the biomass is producing 50% of the oxygen on the planet and respiring about 50% of the carbon dioxide. And it's all microbial. And viruses kill about 20% of that biomass every day. So if I put my oceanographic hat on, then one of the things we have to think about, we have to recognize that viruses are major drivers of geochemical processes on the planet. And so they're very, very important in things in terms of atmospheric nutrients, or sorry, ocean nutrient cycling and atmospheric carbon, for example. And so, so viruses, if we think about it, they actually catalyze the movement of nutrients. So what we've termed the viral shunt, in fact, viruses move material from living particulate material into dissolved organic material. And in fact, that represents about 25% of the carbon fixed by photosynthesis in the world's ocean actually passes through the viral shunt. So it's really, really important in terms of our understanding of processes that are going on in the ocean. We really can't understand these processes unless we start thinking about what viruses are doing in those systems. So that's kind of one of the hats we wear as well. So we spend a lot of time in my laboratory trying to measure those impacts, trying to tie viruses into those geochemical cycles. But the fact that we find all these viruses out there leads us onward. So we certainly, hopefully, I've convinced you of point two. There will be a quiz at the end. There's only 3 points you have to remember. The second one is that they're major players in these global geochemical cycles, and hopefully it makes an intuitive sense why they are. But you know, the obvious question is, well, what are all these viruses? And so what we do is we take a variety of different methods, but what we do most of the time is we go out into the ocean, we collect a whole bunch of water, usually 50 to 200 liters of water. We take that material, we pre-filter it to remove the larger organisms such as bacteria and protists, and then with the viral size fraction we concentrate with tangential ultrafiltration and concentrate the viruses down. Then we can even concentrate them more by ultracentrifugation, etc. In a recent expedition that we were on, we just did our more than 1,000th concentrate of viruses in the world's oceans. So we have ultra-filtered more than 100,000 liters of ocean water. And we archive all that. So we actually have all of these concentrated virus communities are archived, so we can go back and we can look at those over time and space. And I'll give you some examples of what we can do with those kinds of samples. So I'm going to just give you a couple of snippets here. Some metagenomics. I'm going to talk a little bit about the marine DNA virome and the RNA virome. I'm going to give a couple of just highlights of some of the examples to kind of hopefully help you get your head around about just how much diversity there is. So a collaboration we did with Forrest Rowers Group at San Diego State University. We provided a large number of samples from our concentrates. We took 56 virus communities from the Arctic Ocean. We pooled them together into a single community for analysis to get rid of spatial and temporal variation. We took 85 communities collected off the British Columbia coast over 8 years, more than 40 communities collected in the Gulf of Mexico, and then we had a single community from the Atlantic off of Bermuda. And when we started looking at those, obviously I could give a whole talk on these kinds of data, but I think the one thing that kind of summarizes all this up, so this was high throughput 454 sequencing. We ended up with just under 2 million reads from those samples. 90% of the sequences had no recognized homologs in GenBank. And so I think we talk about the great diversity of bacterial communities in the ocean, for instance, the Craig Venter kind of stuff. And the difference is, and this is how we always know we have viruses and we're not contaminants, because 95% of the hits that you get when you look at bacterial diversity do have hits in GenBank, right? There are homologs. You might not know what all the functions are, but there are homologs. When we look at viruses, even when we do longer reads, Sanger sequences and things like that, it's always over 70% of the sequences that we pull down don't have any recognizable homologs in GenBank. So it's a massive amount of diversity that we don't know. Alex Cully, a PhD student in my lab, for his PhD thesis decided to look at RNA viral metagenomes off of British Columbia. And again, we find that typically these circles here represent, I should have just looked at the two sites that we looked at, Jericho Pier off of Vancouver and one further up the coast about 100 kilometers in quite a pristine environment we call SOG or Strait of Georgia. And when you look at these two, again, in the one case, more than 80% of the sequences had no homologs in GenBank. And the other one, it was only 61%, and that's because if you look at that bright blue area, that's something called Marnaviridae, which is a new family of viruses which was based on a virus that we isolated from the ocean, the first picorna-like virus, which infects a protist. And because of that, we were able to get a fair amount of coverage. Interesting enough, those sites are only about 100 kilometers apart, and there was virtually no overlap between even the families of viruses that we found at those two sites.